In this chapter, we're going to take a look at invertebrate species. Invertebrates are a type of animal, but in many cases, they have a unique look to them, and they can appear very plant-like in some cases. And an example of that is what we're looking at on this particular slide. So to start off, as we talk about invertebrates, what we're meaning by this is these are animals that are going to lack a backbone. Typically, when we think about animals, in our minds, we're visualizing vertebrates. So those are animals that have a backbone made up of vertebrae. These are not going to have that, but these are animals that are very diverse. There's a lot of them. And in fact, this is going to be about 95% of the animals. So there are a lot more invertebrates than there are vertebrates. Altogether, we have about 32 phyla. We're definitely not going to look at all 32 in this chapter. We're going to look at a subset of those, and we're going to talk about the characteristics of just that subset of animals. If we start out, though, with a look at a phylogenetic tree of the animal kingdom is monophyletic. So by saying that it's monophyletic, we do have one common ancestor that it's thought that all animal groups diverged from. And we're also including all of the diverged descendants from this ancestor. That ancestor, as was mentioned in the last chapter, is thought to have been a flagellated protist. And that flagellated protist is thought to be similar to what we call choanoflagellates. So if we look at the groups on this slide, in this phylogenetic tree, most of these are going to be invertebrate species. And we're going to look at many of these different ones that you see listed here. If you look at the first branching point that we have, or the first thing that really breaks this down into two groups, that's this part here. And this has to do with the evolution of true tissues. So we're going to look at the first group that branched off, which is the Porifera group, and these don't have true tissues. So this will be our one group of animals, or our one phyla of animals that does not have true tissues. So if we start looking at this phylum, this is phylum Porifera, and this is the sponges. The sponges are unique, first of all, because they don't have true tissues. So when we talk about tissues, we're talking about specialized cells that work together for a specific function, and they're also separated off by membranes. So we're not going to have that in this case. And when you look at sponges, they look very plant-like. So these are sessile, meaning they're not going to be something that moves around. They're non-moving. They're typically going to be anchored in some specific location. And in addition to that, these are going to have asymmetric shapes to them. So in the last chapter, we talked about radial symmetry and bilateral symmetry. In this case, they don't have either one of those. They're asymmetric. They don't have really a top, bottom, right, left, front, back, any of that. So these are considered to be the simplest of all of the animal groups. Now, these are what we call suspension feeders. And as suspension feeders, that means that they're going to basically be filtering water and then consuming the plankton and small animals, bacteria, and things that you have inside that water. Here we have an example of the structure of a typical sponge. When we look at a sponge, they do have a central cavity in the middle, and that central cavity is what we call the spongiocele. In addition to that, they have a large opening at the top, which we're going to call an osculum. What's going to happen here is that you're going to have little pores all over the body of the sponge, and it's going to draw water in. So the water is going to come in through these pores, and then eventually it's going to flow up and out the osculum. When it does that, we have specialized cells, which are lining the inside of this internal cavity. And these specialized cells have a lot of flagella on them. These flagella have a sticky mucus on them. So if there's any small animals or bacteria plankton in that water, they're going to trap that and then they're going to ingest it by phagocytosis. So what's going to happen here is that this is a feeding cell. So this whole cell is what we call a choanocyte. And if you notice, we have a lot of those cells lining the internal spongiocele there for us. We also have an outer layer. So we have an epidermis. We have some other cells in here that have odd shapes that are called amoebocytes. And those amoebocytes, in this case, can take on a range of functions. They're known as amoebocytes because they do have pseudopodia that makes them look similar to amoebas, the protist amoebas. But they're responsible for producing what we call spicules. And that's the yellow structures that you see here. These can give sponges a very rough shape because those spicules are going to be made up of silica, or in some cases calcium carbonate. And so when we think of sponges being very rough and sometimes rock-like, it's because of those spicules that they have inside. There's also a gel-filled matrix that's really inside all of this area here, and that's what we call the mesohyal. Okay, so this is the general structure of the sponge. They are suspension feeders, or filter feeders, we could call them as well. The water is going to be drawn into the spongiocele through all of the little pores that line the outside of the sponge. They are going to filter out any food particles using the flagella that are part of the choanocytes, and then the water that's been filtered is going to exit through the top out the osculum. Now, if we talk about the reproduction in sponges, for the most part, these are going to be hermaphrodites. So when we say that these are hermaphrodites, we're saying that we have both male and female reproductive structures, or we can produce both male and female reproductive structures. That's generally going to be the case here. Typically, they alternate. So one period of time, they will produce female gametes, and then later they may produce sperm gametes. So they're not going to be doing this at the same time, and the reason for that is that this way they can cross-fertilize. And that cross-fertilization is going to be important for genetic variation. So we have eggs that can be produced, and then the sperm are typically going to be modal. So those are going to be transported in the water, and they can then fertilize a nearby sponge, so that we do get that cross-fertilization. In the case of the zygotes, once we do get fertilization take place, the zygotes are going to be flagellated. And since they are flagellated, they can swim to a new location, they can settle, find a suitable substrate, and then they can anchor themselves there and grow into a normal sponge structure. So those are our sponges. That's our simplest of all of the animals. Those are the only ones that do not have true tissues. So we want to make sure that we know that these do not have true tissues. Now, if we go back to our phylogenetic tree, we said that Porifera, those are basal animals because they branch off very early in all of this. Besides Porifera, all of the other animal groups, they do fall into this clade called eumetazoa. Eumetazoa means true tissues. So all of the other animals are going to have true tissues. And what we mean by true tissues is that these are specialized cells. They'll be grouped together. And separated by membranes. And this is separated from other tissues or other cells by membranes. So all of the other animals are going to fall into this eumetazoa category. We're going to look at the first group that branches off from that, which is this one here, which is called Cnidaria. So when we say this, it's a silent C, and the Cnidaria group is going to be what we typically call jellyfish. We have what we call hydras, which is the one on the very bottom here, and then we also have anemones. So these do have true tissues. It's very simple tissues when we're talking about Cnidaria. This is a very primitive group. These are, some of them are going to be modal, some of them are going to be sessile, meaning they're not going to move around, they're going to be anchored. And we'll have different, some that actually can do both, some that have some stages where they're modal and other stages where they're sessile. Now when we look at these, they are simple organisms. They're going to be diploblastic. And they also have radial symmetry. So by radial symmetry, they have a top and they have a bottom, but there's not going to be a front or a back to them, just the top and the bottom. So if we go through and we talk about their general structure is that they have a cavity. So this central cavity inside with one opening. And these are going to be carnivores. So the last group that we looked at, the Porifera, those were filter feeders. These are going to be carnivores. They're consuming and trapping actually small animals and protists. If we take a look at the body structures that we have for this group, this is the first one here, and this is what's known as the polyp. The polyp is the sessile form. So this is the form that's going to be anchored to some type of substrate. So it's not going to be moving around. Notice that with this one, we have all of these little tentacles at the top. And what those tentacles are going to do is they're going to wave around in the sea. And if prey comes along, they're going to trap that prey. If they trap the prey, then that prey can be kind of forced down into this internal gastrovascular cavity. So we do call this a gastrovascular cavity. And what happens in this cavity is that there are going to be digestive enzymes that are secreted from the cells that are all around the outside here. So these enzymes are going to be secreted into the internal gastrovascular cavity. They will digest whatever organisms have been forced down there into that cavity. And then any leftover waste and water can exit back up through the top, and the nutrients will go back and be absorbed into the wall of this. So since it's one opening at the top, that one opening, that single opening, is going to serve as both the mouth, and it's also going to be the anus. So this is where the food comes in. It's also where the waste are going to exit from. Now this group is going to, the groups that have the polyps, a lot of them are called hydras and also the anemones. The other structure that we have looks like an inverted polyp. This is what we call a medusa. The medusa is the modal form, so these are ones that actually move around in the water. And notice that they also have a single cavity inside. That's their gastrovascular cavity. They have one opening at the bottom. This is going to be, again, both the mouth and the anus. So food will come in and the waste will exit from this same opening. And they have a lot of tentacles that are going to wave down around the bottom as well. And the purpose of those is to trap prey and then force it into that gastrovascular cavity. Now these jellies can move around because they can use basically the water that goes into the gastrovascular cavity. They can contract that and then they can open it back up, and that's going to cause them to have some movement. It's not a real good directive movement, but it is going to move them around. So we say they're modal, but it's more drifting than anything else. The name of this group, Cnidaria, comes from these specialized cells which are called cnidocytes. And we see that right down here on the bottom. These specialized cells contain this organelle called a nematocyst, and that nematocyst is able to shoot out of the cell and sting some prey. In many cases, it contains a poison or a toxin that may paralyze that prey. So this is something that's very important for feeding. Now, not all Cnidaria are going to have this, and not all medusas are going to have this, but it is fairly common for them to have it. The Cnidaria are going to have some simple nerves, These simple nerves are going to form a network that really extends around the body. These do have radial symmetry, and those nerves are going to be dispersed all around the body. So this is going to enable them to respond equally to any sensory input that they get from any direction. So again, they're not going to be organisms that have a front and a back to them. They can respond equally on all sides. Now, if we start talking about the individual clades or groups of the Cnidaria, the first one is the hydrozoans. So the hydrozoans are going to be a group that's going to alternate between polyp and medusa forms. So if we take a look at the general life cycle of hydrozoans, or what we kind of commonly call hydras, these will have a colony of polyps. So they're going to live together, and those polyps are going to have two different types. We have feeding ones, which have the normal polyp structure to them. They have the tentacles at the top that are going to capture prey. And then we also have reproductive polyps. Those reproductive polyps are going to separate off from that colony of polyps, and when they do that, they form a medusa that contains either the female gonads or the male gonads. Those will produce the egg and the sperm, and then the egg and the sperm, if we get fertilization take place, those are going to form a zygote. That zygote is going to develop into a larva, which we call a planule. Now this larva is going to have a lot of cilia on the outside, so this is going to enable it to move around a little bit. And since it can move around a little bit, it's going to make its way to some type of anchoring substrate. And once it gets there, then it can develop and grow into that colony of polyps that we started with. So that's the general life cycle of the hydrozoans. So hydrozoans, that was the first group, or the first clade that we have. Those are going to have both a polyp form and a medusa form. The next group is going to be what we call the anthozoa, or anthozoans. And the anthozoans, these are characterized by what you see on this slide, which is in that picture, which is the anemones. This is also going to be the corals. Many of them have symbiotic relationships with algae. So these are a very important part of the coral reefs. The next group is what we call the scyphozoa, or the scyphozoans. These are what we typically think of when we think of jellyfish or medusas. So these are the jellies, predominantly medusas. Not every single one of them is, but most of them are. So these are going to be modal species. And then the last one is the cubozoas, or the cubozoans. And these have a box-like shape to their medusa. So the medusa is going to look a little bit different from the other groups that we've seen. Some of these are highly toxic. The sea wasp is a good example of these. These you don't tend to see washed up on the shore as much as you do the jellyfish, which come from the scyphozoans. Okay, so that's the look at the Cnidarias. These are the simplest animals that we have within that eumetazoan clade.